Good afternoon, everyone. My name is Andrew Buultjens and I'm a postdoctoral researcher over at the Doherty Institute just across the road, and I'm in Tim Sneer's laboratory. And it's my pleasure this afternoon to be moderating the Rapid Poster Talks. And it's been a terrific IMUC-III so far. I've had a great time. I can see everyone's got a smile and they've enjoyed coming together to share our passion in Mycobacterium ulcerans. We've had some really great talks, some long talks of about 20 minutes and some shorter ones. But now we've got to shift gears and go for some really short, punchy, rapid-fire talks. And this is a real light-hearted sort of fun part of a conference, and we've probably all seen lightning talks or elevator pitches before at a conference. I've certainly seen a few, and my favourite method to run them that I've seen is to have a round of applause, to clap when the speaker has gone over time, to sort of clap them off the stage, because we're such a supportive research environment. So the way that's going to work is everyone who has a poster, I think we've got 9 speakers here, they've got to come up and give a 1-minute spiel to give us a feel for their poster, and then there's a break later with drinks, so you'll have a chance to go and talk to those people and see their posters. So it's to whet your appetite. And so we're only giving them 1 minute, and if they go over their minute, what's going to happen is I'm going to start clapping, and what I want everyone in the audience to do is to join in and have a round of applause. So let's give it a test. Okay, so speakers, if you hear that round of applause, you know your time is up. Drop the mic and pass it to the next person. So what I think, suggestion from Rachel just a moment ago, which kind of sounds fun, is what we'll do is we'll get all the presenters to actually line up on the side of the stage here, if they could stand in position. If that's okay. Then there's less time between handover as well. And lastly, I'll just say that there actually is a prize for this session for the best talk and the best poster. That's $100. That's been supplied by Australian Society for Microbiology, so thanks for them. Okay. So first up we've got Michael Malou, who'll be speaking about investigating the role of foxes in Mycobacterium ulcerans transmission in Victoria. Michael, if you can stand up here, take position. Don't start yet. I'll give you the signal.
Test.
Yep. Okay, Michael, your minute starts now. Take it away.
Thank you, Andrew. From this morning, you've heard that the epidemiology of Buruli ulcer in Victoria has not been fully understood. The case numbers are increasing and the disease is being transmitted to new areas. Possums are recognized as reservoirs, and mosquitoes have been proposed to contribute to transmission. We know that possums are quite focalized, so they travel very short distances and may not fully explain transmission of the disease to new areas. Previous environmental sampling has shown high prevalence of Mycobacterium ulcerans in fox feces, and we think foxes might contribute to the transmission of the disease to new areas. So we're currently...
Sorry, Michael. You've run out of time there.
One last one. So our study has done quite a bit of surveillance of Mycobacterium ulcerans in fox samples using fox control activities. Thank you.
Excellent. Thanks, Michael. That's 2 rounds of applauses. That's not good. Okay, so next up we've got Bridget McNamara, and she will be talking about possum faeces heralding the emergence of Buruli ulcer in the Australian urban centre. Take the microphone, don't start yet.
Okay.
Your time starts now.
Good afternoon, everyone. As you've heard a lot today, possums are a key reservoir species for Buruli ulcer in Victoria. And as we've seen in Geelong and Belmont with this higher co-location that Professor Johnson showed, our study described the emergence of the human cases in previously non-endemic areas of Geelong. And we looked at the distribution and timing of human cases in relation to emulcerans positive possum faeces. through excreta surveys done by CSIRO colleagues. And the main finding is we saw that clustered human cases were within 200 metres of positive possum faeces, and they were emerging up to 3 years prior. So in areas where there were no human cases, we could see the infections in possum populations several years beforehand. And this offers a window of opportunity for both targeted education and awareness for community and clinicians to improve treatment, and also an opportunity for research areas if we're needing areas that are likely to be emerging. Thank you.
A minute is not a lot of time when you're trying to talk about your poster, is it at all? Very unfair. Okay, next up we've got Serges Chachawing. Apologies if I got that wonderful surname of yours incorrect. If you could please take the stage, just one moment, and your time starts now.
Thank you. A traditional collection method for Buruli ulcer sample like swab can be painful for patients. This project aims to evaluate the use of sponge alternative sample source for BU diagnosis. For this purpose, active case search was performed in 2 health districts in Cameroon. For each suspected Buruli ulcer cases, swab and sponge were collected from skin lesion and analyzed by PCR. Our findings show that there were high detection rate in sponge for Mycobacterium ulcerans compared to swab. And moreover, the sponge show substantial agreement with swab, suggesting that the sponge can be used for BU diagnosis. By reducing patient discomfort and improving diagnosis accuracy, sponge can be used for BU diagnosis. Thank you.
All right. Next up, we've got Jessica Porter, who's also from the Sydney laboratory, where I'm from. And she'll be talking to us about a single copy gene target for Mycobacterium ulcerans quantitative PCR.
Okay.
Take it away, Jess.
I will stay under the minute, I think. So my poster out there is discussing work we've done on a single copy gene target for M. ulcerans, which I guess everyone's very familiar with the multi-copy target 2404, so we've done some comparison to that on qPCR, as well as transferring both assays onto a digital PCR platform, which had some advantages over qPCR. So that isn't...
Well, that's our first presenter within the minute. Congratulations to you, Jess. Next, we've got Emma Sars-Lopez talking to us about drug acceptability studies of Mycobacterium ulcerans clinical isolates in Benin.
No, it changed, but...
Oh, you've changed. Okay.
It's
Do you want to
fine.
quickly say what you mean?
Yeah, it's okay.
On that?
I will.
Yep.
Yeah.
All right. Real curveball here. So your time starts now.
Okay, thank you. Well, so it changed. As you can see, I'm going to present a study on the bacterial clearance rate in Buruli ulcer lesions from patients in the beta-lactams for Buruli ulcer study. So this is a study to demonstrate the to show the bacterial clearance rate in patients in the context of a clinical trial. The beta-lactams for BU wants to evaluate the possibility to shorten the treatment from 8 to 4 weeks. So we are comparing two treatments, the current treatment of rifampicin clarithromycin of 8 weeks versus the 4 weeks by the coaddition of amoxicillin clavulanate. So we are doing this study in POVE treatment center. We studied in December 2021, and we take 5 samples for Buruli ulcer diagnostic and also to test the viability assessment of Mycobacterium ulcerans. So far, we have processed samples from 50 patients, and we observed that more or less 70% of patients showed viable bacteria. Okay.
I felt like you were just about to wrap it up then too. I'm sorry about that. Okay, next we've got Santiago Ramon Garcia, talking about model-informed development of antimicrobial combinations in Buruli ulcer. Your time starts now.
I'm actually presenting on behalf of Umberto Villani from University College of London, with whom we are collaborating on a modeling project. The study is within the beta-lactams clinical trial in West Africa, and what we are doing is combining in vitro methodologies with modeling and simulation methodologies to predict treatment shortening. So the modeling activities that we are doing actually can inform on which combinations and which treatment can clear, in this case, a culture, but we hope to translate into clinical treatment shortening.
Good work. That's impressive. Excellent. That was a good clap you got then. That's a slide. Okay. You have to do it again. Okay. The next speaker is Shashinta Wajagunasekara, but unfortunately he will not be here today. So Shashinta's prepared this in advance for us. It would be rather embarrassing if it goes over a minute, I suppose.
Our study, Understanding the Pathogen and Microbiome Variation of Buruli Ulcers, explored the Buruli ulcer microbiome of Australian patients through metagenomic sequencing. In this study, we sequenced DNA extracted from 24 pre-treatment ulcers, 3 treatment and 3 post-treatment ulcers. Some key findings of this work are that severe ulcers contain higher microbial diversity, anaerobic bacteria dominate most ulcers, and fungal species are more present in ulcers undergoing antimicrobial therapy. Antimicrobial resistance gene profiling indicated resistance genes associated with bacterial genera such as Corynebacterium, Bacillus, and Klebsiella. To learn more about this work, please visit our poster. Thank you.
Okay, second last speaker in this session is Yvonne Asante Kouafo. She'll be in tomorrow. Okay. Right, so we'll skip that speaker. And the last one is Andrew Buultjens. How do you say that? It's another hard name. All right, that's me. So one of those weird opportunities to chair my own talk. So, yeah, I've got full autonomy here, don't I? It's overwhelming. All right. Okay, so Buruli ulcer is a devastating disease with a 4.5-month incubation period. And because it's so long, it often means patients have a hard time recalling where they were infected with this pathogen. And that's a big issue for epidemiologists working in this space because they can't figure out where the bulk of transmissions are occurring. And that's an issue because that information is very important to direct public health interventions to where they're needed. So what we've done is we've used the relationship that we've seen between genotype and geography with this pathogen to train a neural network to predict where patients acquired their infections. And we've trained this on a large number of bacterial genomes, and we've shown that it can accurately predict areas of elevated risk. So this new approach empowers public health officials with the ability to make better risk assessments and therefore to direct public health interventions to where they're most needed to have the best impact on disrupting transmission of Buruli ulcer. And 5 seconds to go. Okay, so thank you very much for that. Can I have a round of applause to everyone for the entire session? Okay, so I think we're now moving on with the final talk. Maybe don't run away too quickly. So we've got 3 minutes remaining to the next speaker. Perhaps we'll open up to a quick question.
The person that said that you tested for viability of Mycobacteria after, was it 4 weeks of Rif-Clary and Amoxiclav? Did you say 70% were viable at, what was the time point at which they were still viable? Was that at the 4 weeks?
We are taking samples at different times. Yep. So it's more or less, depend, but the idea was 1 week, at 2 weeks, 4 weeks, 8 weeks, and 14 weeks for patients who have the lesion healed. So after 28 days, most of the patients have no viable bacteria. Yes, this is how it. This was your question?
That answered my question. Thank you.
Okay.
Stacey? Yeah.
Less than 3 minutes, we'll start clapping. The gentleman from Deakin University isn't here, but I hope that, Dan, I'm going to put you on the spot. Interesting to see you looking at the microbiome of the lesion. Are you looking at microbiome of black possum lesions as well and seeing if any...
Just clinical samples at this time for those on Zoom. 2 minutes left. Any more questions? No? All right, we'll leave it at that.
Okay, thank you. Let's move on to the last session of the day. And we saved the best until last. I'm just going to wait 1 minute just to make sure we have Francoise queued up correctly. So it's my great pleasure to welcome the AV technician to the stage. Thank you.
Do you hear me?
Oh, no, we're just getting you set up, Francoise. Here we go.
Okay.
Yeah, very much. Francoise, I hope you can hear us. I'm about to introduce your presentation and introduce you. And then you'll have the opportunity to share your slides and begin your presentation. So it's my great pleasure to introduce to you someone that perhaps needs no introduction to many of us, but Professor Emeritus Francoise Portels from the Institute of Tropical Medicine in Antwerp. Dr. Portels is Professor Emeritus at the Institute of Tropical Medicine in Antwerp, Belgium, and the Free University of Brussels. Her 40 years' experience in mycobacteriology, mainly on Mycobacterium ulcerans disease, and in the field, notably across many countries in Africa, have enabled her to study the clinical aspects and the epidemiology of the disease, as well as the environmental reservoir of M. ulcerans. She was the first to succeed in culting M. ulcerans from the environment and hypothesise a possible role for insects in transmission. Now that very much understates Francoise's achievements in this field. She has trained innumerable people, many of you here, to go on to develop your own successful careers. She's a mentor to many, a friend to many of us. Francoise, we're delighted you could join us, even if it is remotely. Better some Francoise than no Francoise here, so thank you for joining us, and the floor is yours, and we look forward to your presentation.
Thank you. Can you hear me?
Yes, we can hear you.
Okay. So first of all, I would like to thank you, Tim, for your introduction. You have done and you have said everything. And I would like to pay credit to two old Australian friends who enabled me to know your beautiful country and to know more about Buruli ulcer in your country. May Smith and John Heyman. But first of all, a few words to tell you how I got involved in Buruli ulcer research. It was in 1970 that my director, Professor Janssens, who was the person who discovered Mycobacterium ulcerans in Congo in 1942, that he sent me to Congo to search for environmental reservoir of Mycobacterium ulcerans. And I had the privilege to meet there for the first time Dr. Wayne Myers, who was working in Kuvuru in Lower Congo. He was a nephrologist and had already discovered several cases of Buruli ulcer and had made a beautiful map, as you can see here on this slide. So during 4 years, I have collected a lot of environmental specimens and isolated mycobacteria from them. It was the subject of a PhD thesis, and the first conclusion was that mycobacteria were present everywhere in the environment. I had to build my own lab of bacteriology, of mycobacteriology there in Congo, and the mycobacteria were identified later in Antwerp. And we discovered that there were seasonal variation, that the most frequent species were Emergium complex, Terre, Gordone, Fortuitum, but also more than 10% were new species. And some of these new species were only present in Buruli endemic areas, but unfortunately no one culture of Mycobacterium ulcerans. So in 1992, I was contacted by Dr. Guy Denon from Benin, who explained me the situation of Buruli ulcer in his country, and together with him, Dr. Myers and Sister Julia from Zanianado, we decided to study Mycobacterium ulcerans disease in Benin. We, as you said, Tim, we analyze the clinical, epidemiological, microbiological aspect, the social aspect too, and we collected, of course, environmental specimens. Thanks to the discovery of Ruth Ross in 1997 of the IS-2404 element, insertion element, we were able this time not only to study these mycobacteria by culture, but also by PCR. And in 1999, we had this hypothesis that maybe water bugs could transmit the disease and are passive vectors of Buruli ulcer, of Mycobacterium ulcerans. And during 12 years, we have isolated a lot of mycobacteria and have found several positive PCR specimens, aquatic insects, fish, and also some other, like mollusks, for example. But the terrestrial mammals that we studied were all PCR negative. And in 2008, finally, we were able to cultivate Mycobacterium ulcerans from the environment. So the first lesson was that we have to be very patient to cultivate Mycobacterium ulcerans from the environment. And it was not so easy because, as you know, there are some other mycobacteria, and we have to get rid of these mycobacteria. And that's the reason why we succeeded in cultivating Mycobacterium ulcerans by first culture in Bactec and pre-passages in mouse foot pads to get rid of all these environmental mycobacteria that grow much faster than Mycobacterium ulcerans. And it was isolated, that's funny, from a water bug that is not able to bite humans, and also called the Jesus bug because it moves rapidly on water surfaces. So the first lesson is that we have to be very patient, and that cultivation remains a tedious procedure, but detection of viable bacilli is essential. So never ever give up. A few words about the transmission, which remains poorly understood. As you know, the various routes may be different according to the geographic region. The major mode of transmission is probably via Buultjens, but aquatic insects and other insects may play a role. In Africa, it remains controversial, but now in Australia, it's clear that mosquitoes, Aedes notoschriptus, play an important role in the transmission. And we also have studied the role of trauma, and a few words about the trauma. On more than 1,000 laboratory confirmed cases, we were able to identify 29 patients who really clearly spoke about a trauma. What is interesting on this slide is that most of these trauma were non-penetrating trauma. Blunt traumas, fall, hit, blows, and also massages. So, what are the different possibilities concerning BU after trauma? Is that the skin surface may be contaminated by Mycobacterium ulcerans, and that the trauma may introduce ulcerans into the dermis or subcutaneous tissue. The second is that insects may be contaminated and may transmit the disease, as it was confirmed in Australia by me and co-workers. But don't forget that trauma also may be important because they may activate a latent infection. So my second lesson would be that research should focus on mycobacterial latency. It is well known for TB and leprosy, but very bad documented for Buruli ulcer. And there are some arguments in favor of activation of latent infection. For example, in Africa, you have BU in people older than 60 years old, and these people don't go to the swamps anymore and so on, and maybe it's a reactivation due to an immunosuppression. Another possibility is that that some patients were diagnosed in non-endemic areas and coming from endemic areas where they were infected, and also that trauma may stimulate the development of Buruli ulcer. And there is a beautiful example about that, published in 1970, is a Nigerian physician who scraped his elbow on the bathroom mirror of two months after arriving in New York and developed a chronic ulcer diagnosed as Buruli ulcer. I don't think that the mirror was contaminated by Mycobacterium ulcerans. So future research should really focus on the role of trauma and latency in Buruli ulcer. A few words about the taxonomic position of Mycobacterium ulcerans. In 2012, Doherty published a paper where they considered that Mycobacterium ulcerans and mycolactone-producing mycobacteria were all coming from a common variant, marinum progenitor, and that they are all belonging to the same species with different ecovar. But clearly, these different mycolactone-producing mycobacteria are quite different from a phenotypic point of view, and also they are pathogens for different hosts. So I think that my third lesson would be that all MPM classified under single species may be confusing from an epidemiological point of view and a medical point of view. And one example is this publication from 2013 where Mycobacterium ulcerans, so-called Mycobacterium ulcerans, was cultivated, demonstrated in southern Louisiana. It was an MPN, but not ulcerans sensus stricto. So it may be confusing also from a medical point of view. Another point that worries me is the differential diagnosis of Buruli ulcer. It's not so easy. And one of the proof is that so-called new foci of Buruli ulcer were recently described, but some of them were not confirmed by lab test. The clinical feature was not typical, and when histopathology was done, the histopathology was not at all specific for Mycobacterium ulcerans. Only PCR was used. And unfortunately, there are also some fraudulent results, and we'll have some proof of that also. So the fourth lesson would be that strict criteria should be applied to confirm new foci of Mycobacterium ulcerans disease. And if it is confirmed by PCR, please participate in external quality assurance. And it is also important to confirm by at least 2 different laboratory tests, like for example, culture and PCR, histopathology and PCR, direct smear examination and culture and so on. Because as you know, well, case finding is not a treasure hunt. And now I would like to say a few words about the scientific quality of the publications. The first publication on Buruli ulcer were outstanding, and I give you here a few examples. Of course, the famous paper of McCallan and co-workers, but also the papers from the Uganda Buruli group. All these papers were outstanding, and I just encourage young people to read and to reread these papers because they are a gold mine of information. But since the 90s, there is an exponential increase of Buruli ulcer papers, but not all from outstanding quality. And because there is this culture of publish and perish that was described quite rightly by Wright in 2024 nor recently. As a result, an increased productivity, but also an increase of bias, an increased use of artificial intelligence, paper mills, predatory journals, and also fake papers. So last but not least lesson would be that the quality of publication should trump quantity and avoid the pitfall of publish or perish culture, maintain a high level of research integrity and self-criticism, be aware of our own bias, be open to correct errors, including honest errors. And by the way, John Heyman was able to do that because he published a paper who described that he was not right in some hypotheses, so that's very nice, and promote especially multidisciplinary studies. And there is a beautiful example of a marvelous recent paper published, and I am pleased to tell you that it is the paper on mosquitoes in the transmission of Buruli ulcer in Australia. So in conclusion, I would say that there are many other lessons that we can learn about Buruli ulcer after half a century. But just, I would say, like Ioannidis said recently, is that my main conflict of interest was trying to be an honest scientist. This means that I was probably biased and also often wrong, but not totally resistant to the possibility of getting corrected, even if I am now over 80. So I would like to thank all the PhD students that have performed work on Buruli ulcer in the Institute of Tropical Medicine, is thank them that the work was done, and that we have published over 100 papers, published peer-reviewed paper on Mycobacterium ulcerans. And finally, I would finish to thank my successor, Professor De Jong, because thanks to her, there is a future of Buruli ulcer studies in the Institute of Tropical Medicine, and as it is evidenced by a recent paper of Schudy and co-workers on volatile organic for the diagnosis of Buruli ulcer disease. Thank you very much, and have a nice drink.
Thank you very much, Francoise. Would you be open to taking some questions from the floor?
Yes, no problem.
Yeah. Does anyone have any questions or comments or reflections they might want to raise while we have Francoise out of bed early and online? All right, we have a question from Paul or a comment from Paul Flood. Paul, I'll pass you the microphone so that Francoise can hear you. Maybe someone could do microphone shuttling for me.
Thank you. Francoise, is Paul Flood from Phillip Island, whom we met...
Good. Good afternoon, not good morning, good afternoon.
In the early 90s. It's very good to see you looking so young. Fortunately, you can't see me, I'm grey. Secondly, I'd like to ask you, is that the picture of a eucalyptus tree behind you? Because it looks very much like an Australian tree.
Yeah, that's right.
Thank you very much.
Look,
Our koala.
Behind me, a small koala.
Yeah, well, thank you so much. And as you know, koalas on Phillip Island had ulcerans lesions. And thirdly, related to your question about potential trauma inducing latent infection, what do you think about supporting an ethics proposal to get a cohort of people and inoculate them with a needle in an endemic area to see whether they may have latent ulcerans in a subcutaneous tissue and see whether they develop disease? Create artificial trauma in a controlled cohort. Do you think that would pass ethics approval?
I don't think so. Well, in my opinion, it may be dangerous from an ethical point of view. But what I think is that I have seen that in my own files on Buruli ulcer in Benin, is that we really need to use social sciences, at least in Africa, to interview people to know more about trauma, because we had only 29 patients. I'm sure there were much more, you know. And what is amazing is that some of these patients with trauma developed osteomyelitis. So I don't at all know if there is a relationship between the trauma and the reactivation of latent infection, but it worries me. So it would be a good suggestion, but I think it will be a problem from an ethical point of view. You have to see with your own ethical committees. It's nice to see you. Well, to hear you, Paul.
We have Paul Converse.
Yes. Hello, Francoise. Very nice to see you again. You were talking about the massage-related cases, and it reminded me that at least in leprosy, there have been reports of disease coming about after tattoos. And as I used to have to explain to my students why people got tattoos, now I ask my students to tell me why people get tattoos. However, with tattoos being so prevalent now, and if there is surface contamination of the skin with Mycobacterium ulcerans, this might be another angle to look at the possibility of trauma inducing disease from a surface contaminated skin.
Yeah, that's right. Tattoos are a source of contamination by mycobacteria. It's well known. Do you know that in China there is a publication from China of cutaneous tuberculosis after tattoos and after acupunctures also? So it is also, all skin effractions may be a source of inoculation of mycobacteria, but maybe also a source of reactivation of a latent infection. And it's nice that to make a comparison between leprosy and Mycobacterium ulcerans disease, because there are some common points. And for example, one point that should be analyzed also is to see if aerosol transmission is a possibility. It was always excluded in aerosol transmission, aerosol contaminated by ulcerans and aerosol transmission of this Buruli ulcer. It's something that is not enough explored.
And just one additional comment. One of the first papers of tattoo leprosy involved 2 American Marines who received a tattoo in Melbourne, Australia.
No, I did not know that. But I have made a conference on all the consequences of tattoos, acupuncture, and all the problems that we do have with some mycobacteria, especially with Mycobacterium chelonae, for example. There are a lot of papers on this subject, so you are right.
Francoise, I'd like to take you back to one of your comments around transmission, where you said the pathogen could enter through cuts and scratches. The evidence we have from laboratory modelling of transmission, and I'm going to look to Professor Jordan's work here and our own work, is that you need inoculation to establish infection, and it is not sufficient to have the bacteria present on a cut or a scratch or on a superficial lesion. You need to, a penetrating can be a micro trauma, but it needs penetrating. Did you want to comment on that?
Yes, you're right. It's exactly the same for leprosy. You know, leprosy, you need also introduction into the cutaneous or subcutaneous tissue or dermis, and it's the same for Mycobacterium ulcerans. You are right, and that was a beautiful paper from Hatter and co-workers. Also, what is interesting is that the number of bacilli that you need does not need to be very great. A few bacilli are enough to get the infection.
Thank you. Are there any other questions for Françoise? It's a pity, Françoise, I don't think you can see the audience, can you? You can only see see me, which is...
But I see you.
Yeah.
And that's enough for me.
That's enough, oh yeah. Fantastic. I'm blushing, yeah, thank you. Any other questions or comments for Françoise before we let her get to her cornflakes? Okay, well, Françoise, we are going to thank you again for joining us, and it has been a delight to hear you and for you to close our first session of IMUC-III. Thank you very much.
Thank you very much for your invitation.
All right, thank you, friends. So that concludes day one, at least the official proceedings. We've got some drinks for those of you that have enough stamina to... Sasha, you can do it, I know. For our Northern Hemisphere colleagues that are feeling a little jaded, nothing better than a glass of red wine to pep you up. So anyway, drinks and some, I think some food perhaps, or at least drinks are out there now. Thank you, everyone, for a terrific start to IMUC-III. Thank you, Françoise.